After sunset on a coral reef, you may find a parrotfish suspended inside a clear envelope of its own mucus. The fish is not trapped, diseased, or wrapped by another animal. Before resting, it has secreted the material and spread it around its body like a transparent sleeping chamber. By dawn, the envelope may be broken, abandoned, or nearly invisible in moving water. The nightly construction demands time and biological material when a resting fish is already vulnerable. That cost creates the central mystery. What danger makes a disposable cocoon worth producing? Early explanations emphasized hiding scent from predators hunting by chemical cues. Later experiments provided stronger support for protection against blood-feeding gnathiid isopods. Neither explanation should be treated as universal proof for every species, reef and night. The cocoon may have several effects, while its primary benefit depends on local threats. Direct observation establishes the sequence, but function requires comparing protected fish with meaningful controls. At dusk, feeding slows, a resting site is chosen, and secretion begins around the body. During darkness, the barrier remains while small attackers search among reef crevices. At sunrise, the fish leaves and resumes grazing across exposed coral surfaces. The answer lies where sleep behavior, parasite pressure, and reef ecology meet. Parrotfishes belong to the family Scaridae within a larger group of wrasses and their relatives. They are best known for fused or closely packed teeth used to scrape food from reef surfaces. Many species graze algae or detrital material associated with dead coral and rocky substrate. Their feeding can remove algal growth, process carbonate, and alter competition across tropical reefs. Not every parrotfish uses a mucus cocoon, and behavior differs among species and individuals. Well-documented cocoon builders include several species in the genera Scarus and Chlorurus. The fish usually selects a sheltered depression, crevice, or position near reef structure before settling. Reduced nighttime activity lowers energy use, but immobility creates opportunities for predators and parasites. Reef darkness does not remove chemical traces, water movement, or contact with roaming attackers. Mucus is a complex biological secretion rather than a simple sheet of water. It contains large molecules that give the material viscosity and allow a coherent envelope to form. Producing it uses resources that could otherwise support maintenance, growth, or ordinary body mucus. The skin's usual mucus layer already contributes to protection and surface function. The sleeping cocoon extends beyond that layer, creating a surrounding boundary with space between fish and environment. Formation can take tens of minutes, though timing varies, and fragile precision is inappropriate across species. The fish releases mucus near the head and works its body within the accumulating material. Fin and body movements help shape the secretion into an enclosing structure. Once settled, the fish reduces movement while water continues flowing around the cocoon. The enclosure is permeable enough for the fish to remain ventilated. It is not a sealed bubble supplying stored oxygen through the entire night. Water still reaches the gills, so dissolved chemicals and tiny organisms are not absolutely excluded. Any protective effect must therefore operate as a partial barrier rather than perfect isolation. If you shine a light across the cocoon, its transparency seems poorly suited for simple visual concealment. Night predators, however, may rely on smell, vibration, silhouette, memory, or combinations of cues. The scent masking hypothesis proposes that mucus slows or modifies chemical signals leaving the resting fish. This remains plausible for some encounters, but demonstrating it requires measuring predator detection directly. A predator failing to attack could reflect absence, satiation, poor visibility, or many unrelated factors. Experiments must compare otherwise similar fish with intact and disrupted cocoons. They must also avoid making cocoon removal itself the source of stress or unusual odor. Another hypothesis predicts that the barrier blocks ectoparasites seeking a stationary host. Gnathiid isopods are small crustaceans whose juvenile stages take blood meals from fishes. These mobile juveniles often emerge at night and can locate hosts resting near the reef. Contact with a cocoon presents a physical and possibly chemical obstacle before the fish's skin is reached. In controlled field experiments, cocoon removal was associated with more gnathiid attacks on sleeping parrotfish. That result supports anti-parasite protection more directly than observations of undisturbed sleep alone. The cue is nighttime parasite activity. The action is cocoon construction, and the consequence is reduced successful contact. Yet one experiment cannot establish identical benefit across every cocoon-building species. Parasite abundance changes with habitat, season, current, host density, and local community. Where gnathiids are scarce, other benefits might contribute more strongly to maintaining the behavior. Where predators use chemical cues, slowed odor release could still supplement parasite defense. Such possibilities remain hypotheses unless tested against specific alternatives. Claims that the cocoon makes a fish invisible, odorless, or immune exceed available evidence. The barrier can tear, and persistent attackers may sometimes cross or move around it. Parrotfish can also awaken and leave when disturbance becomes strong enough. An alarm cue prompts fin movement, cocoon rupture, and rapid escape into surrounding structure. The consequence is immediate mobility, but loss of the night's manufactured defense. Some individuals build another envelope after disturbance, adding further energetic cost. For a grazing fish, nighttime defense preserves the animal that will work across the reef after sunrise. When morning light returns, parrotfish leave shelter and begin repeated scraping bites. A visible patch of algae prompts approach, jaw contact, removal, and swallowing of surface material. The consequence can be cleared substrate and redistributed nutrients, though effects differ among feeding modes. Some species excavate carbonate more deeply, while others take shallower scraping bites. These distinctions matter because parrotfish should not be described as one ecologically identical group. Survival through the night sustains their daytime roles in reef food webs and habitat dynamics. Parasites do more than remove small quantities of blood from an isolated host. Heavy or repeated attacks can impose stress, injury, and energetic costs. They can also influence where fishes rest and how long protective behaviors continue. The cocoon, therefore, connects microscopic nighttime encounters with larger patterns of habitat use. If parasite pressure changes, the balance between construction cost and defensive benefit may also change. Reef degradation can alter crevices, currents, predator communities, and parasite distributions simultaneously. Artificial light may affect nocturnal activity, although consequences for cocoon use require direct study. Warmer water can change metabolism and ecological interactions, but simple predictions would be premature. Human observers often focus on the strange envelope and overlook the ordinary vulnerability it addresses. Rest is necessary, yet reduced responsiveness creates a recurring window for exploitation. Evolution can favor a temporary barrier when nightly survival gains exceed the resources spent producing it. That explanation does not require foresight by each fish before every construction. The behavior can be inherited, flexible, and adjusted through immediate environmental cues. Future research should measure cocoon composition across species rather than assuming one uniform material. If you compare intact envelopes chemically, defensive compounds might explain differences beyond physical thickness. High-resolution nighttime video could reveal which organisms contact, penetrate, or avoid the barrier. Parasite counts should be paired with host condition to show consequences beyond a single interrupted meal. Predator trials could test chemical detection while controlling water flow, light, and fish movement. Researchers also need energetic estimates for secretion, rebuilding, and lost resting time. Long-term identification might reveal whether older fish choose safer sites or build more efficient cocoons. Comparisons between reefs could test whether construction frequency tracks gnathiid abundance or predator pressure. Such studies must distinguish absence of evidence from evidence that a proposed function is absent. An untouched cocoon records no attackers, but it cannot reveal which attackers never approached. An experimentally removed cocoon provides contrast, though manipulation can create its own artifacts. The strongest conclusions will combine direct encounters, controlled comparisons, and measured fitness costs. You can then interpret the envelope as an adaptive barrier without turning it into magical armor. At dusk, the parrotfish still faces a reef filled with moving water and sensory trails. Its secretion hardens no wall and guarantees no safety, but it changes the odds of contact. By morning, the fragile chamber has served its brief purpose and can be left behind. The opening image remains transparent, yet evidence shows that transparency can still obstruct a blood-seeking parasite. Parrotfish mucus cocoons are documented nighttime defenses, with anti-parasite benefit supported experimentally and other proposed functions still requiring careful tests.